Alright, hello biology students. One thing we should cover that, you know, I think it's kind of looked over because we spend most of our effort with transcription and translation is DNA replication. And of course, you might know this as DNA copying, so making a copy of it. And one thing you need to remember is when is this occurring? When is the DNA replicating? And this is something the students kind of forget about. And don't forget about when we talked about mitosis and meiosis, that when the DNA has to double during the S stage of interphase, that's when this is going on. So this video is to attempt to refresh your memory about what happens during the S stage and actually show you the mechanisms involved. So real quick, let me just draw a DNA strand here for you. Okay, DNA double helix. It's going to be unwound, so it looks like a ladder. Okay, now I'm not going to draw different colors just yet. Now here is our nitrogen bases. Remember, with complementary base pairing, A bonds with T and G bonds with C when we were talking about DNA. Okay, and so let me just draw the rest of them here. All right, now we have to double this DNA. Okay, so what's going to happen is this: an enzyme called helicase, okay, is going to bind. Okay, so there's an enzyme called helicase. I always tell my biology students, anytime you see the word ASE, that means it's an enzyme. And what helicase does is it unzips, essentially. Unzips the DNA, or if you want to get really, scientific, it breaks the hydrogen bonds. The hydrogen bonds are what hold these bases together. So it breaks hydrogen bonds. Okay? And so essentially it's going to move this way, the helicase, and the other one's going to move this way. And it's going to create a Y shape. So let me draw the helicase in motion now, essentially. So now we're going to have a DNA strand. And I'm going to draw it really exaggerated, really unzipped, because I want you guys to understand. All right, this is really unzipped. All right, so we got to draw our helicase that's unzipping the DNA. Think of this helicase if you're confused about a zipper on your jacket. If you zip up your jacket, the zipper binds together, and if you unzip it, it splits apart. So the helicase is like a zipper. All right, so there we have our helicase. Let's draw our base pairs. Okay. And of course, we got to have our base pairs over here. Now, there's more advanced videos out there on the internet about DNA replication, but my videos are not intended to go to that degree. My videos are to just get my students a basic understanding of what DNA replication is, keep the vocabulary to a minimum, and mostly just study the function. Alright, so now we have our helicase that is unzipped. Now we're going to add a new enzyme, and I'm going to put it right here. I'm going to put it a little bit right here, okay? And there's going to be one here, and there's going to be one here, okay? Now this is called DNA polymerase. There's that ASE again, which tells you that it's an enzyme. And its function is to, let me get a little bit more of space here. It's an enzyme, and its function is to basically build new DNA. And it proofreads the base pairs. Because remember, if you mess up one of those base pairs, you're going to cause a mutation, so it proofreads it. So essentially, it's going to move down the DNA, and it's going to start adding these base pairs. Now, I'm going to draw the new DNA in this purple-pink color. And so what's going to happen is, as it moves down, it's going to create this DNA, and it's going to follow that complementary base pairing again. as it's making this new DNA strand here, I'm drawing it in like the purple-pink color. Those are going to be the correct base pairs, those A's and T's and G's and C's, okay? And so there's our new strand that was created by the DNA polymerase. And essentially the same thing over here. And there's another new strand of DNA. So now if you see here, we essentially, let me get a different color out so I can emphasize it. There we go. So remember, DNA replication is making a copy. After the DNA polymerase is done, you're going to see there's one new strand of DNA, and here's going to be another new strand of DNA. So now there's two new strands, and now essentially the cell can go along and divide. I hope you found this was very helpful. If you have any questions, leave them in the comments. Thank you.